So before starting today's symposium, I'd like to introduce Emeritus Professor Koichi Yoneyama. The Professor Yoneyama started working at the Utsunomiya University from 1978. And then he became the professor of the Weed Science Center of the Utsunomiya University from 1998. And then he became this emeritus professor from April 2018, and he also became the research scientist at the Women's Future Development Center of Ehime University from 2019 to 2021. And his special research area is first one is chemical communications between plant and living organisms, including angiosperm parasites, mycorrhizal fungi, and rhizobia. And the second one is purification and structure determination biology, active compounds of plant origin, including plant hormones. And the third one is molecular design of biological active compounds. And he is a quite well-known researcher in this field, and he also awarded many awards. In 1991, he was awarded for Young Scientist of Japan Society for Bioscience, Biotechnology, and Agrochemistry. And in 2003, he awarded the Society Award of Japanese Society for Chemical Regulation of Plants. And in 2010, he awarded Society Award of Pesticide Science Society of Japan. And in 2012, he was awarded the Thomson Reuters Research Front Award. And in 2015 and 2016, he was awarded Thomson Reuters Highly Cited Researchers. And in 2019, he was awarded Japan Prize of Agriculture Science and Yomiuri Prize of Agriculture Science. Okay, so now let's please Professor Yoneyama start your symposium.
Thank you. Can you hear me?
Yes.
Okay. Thank you very much for Professor Yokota for your kind introduction. I'm very pleased and honored to have this occasion to give a lecture on our research. Today, I would like to talk about the root parasitic weeds, their biology, and control measures. Next slide, please. Okay. About 1% of higher flowering plants are parasitic plants. They attach to other plants for their survival. Click, please. So, yeah.
So
one click, please.
one click.
Click or.
Okay.
Okay. So parasitic plants can be divided into two groups according to the site of attachment. Some of them attach to the shoot, they are shoot parasite. The other attach to the roots, a root parasite. And also parasitic plants have a different host dependency. Some of them can survive without attaching to the host. They are facultative parasite. The others, hemiparasite and holoparasite, needs to attach to connect to the host for their survival. Hemiparasite contain active chloroplasts, so they have photosynthetic activity, but their photosynthesis is not enough to support their lives. So they have to connect to the host. Holoparasite lack chlorophyll, so they are dependent on the host plant for the supply of nutrient and water. So as you can see on the right, it's the largest flower, Rafflesia, and on the right, also on the bottom, there are many trees, and including sandalwood. Okay, next slide, please. Oh, next, please. Okay, this is a typical shoot parasite, mistletoe. Mistletoe cause severe damage to the forestry production, especially in North Europe and also the America, North and South America. And mistletoe is very popular also in Japan. But in Japan, the damage was not so large, not so intense. Okay, next, please. This is another shoot parasite, Doda or Cuscuta species. They can spread. They already spread everywhere in the world, so they are causing severe damage to vegetables and also to some orchard. Okay, next slide, please. Among these parasitic weeds, there are two important root parasitic weeds, witchweeds, Striga species, and broom rapes, Robanki, Feripanki species. Witchweeds, Striga species are hemiparasite. It means that they have functional chloroplast, but their photosynthesis is not enough to support their life. If you can see on the top, this is a flower of Striga helmontica, and you can see some green parts. It's just very small leaves, scale leaves. And Striga asiatica and Striga helmontica parasitize monocots, so important food crops, including sorghum, maize, upland rice, millet, sugarcane, and so on. And broom rapes, Orobanki feripanki species, are hollow parasite. So they have to attach to the host because they don't have any functional chloroplast, including the Feripanki ramosa, Orobanki curenata, Feripanki aegyptiaca, Orobanki minor. Broom rapes parasitize on dicots, including tomato, tobacco, carrot, sunflower, and others. Next, please. This is a sorghum field in Sudan infected by Striga helmontica. On the right, you can see the pink flower of Striga helmontica. And on the left, you can see the sorghum growing normally. Also, there are some sorghum on the right-hand side, but you cannot see them because the growth of sorghum is heavily stunted. Okay, next slide, please. When sorghum, Striga attach, so many Striga attached to the sorghum, sorghum will die, and also the Striga will die. But before Striga dies, they produce huge number of tiny seed, which remain viable for many years. This is a very important or very destructive factors that once you have Striga, you cannot grow the potential host crop for many years. Next, please. This slide on the map on the left show the distribution of witchweed in Africa. So they are Striga helmontica and the Striga asiatica. And sub-Saharan African countries are infested by these witchweeds. So Striga species infect many of the staple cereals or food crops. And so they are causing very bad damage to the food production. And it was estimated that the striga is a major constraint to crop production in sub-Saharan Africa, causing average yield losses of 40% or sometimes total crop failure. Greatest impact in low input subsistence farms, 17 million hectares infected with striga. So continuous cropping has increased the spread of striga. In fact, these, for example, striga helmontica and striga asiatica became weedy when the large-scale agriculture have been introduced. So they became very bad weed only recent 100 years or so. Control is very difficult. I will explain this later. Next, please. This is another type of root parasitic weeds, broom rapes, Orobanchi and Feripanchi species. These broom rapes have been distributed widely in the world. On the left top, it's Orobanchi cumana. It's a sunflower broom rapes. This Orobanchi cumana parasitizes only sunflowers. On the right, it's Orobanchi crenata. On the left bottom, it's Feripanchi aegyptiaca and Feripanchi ramosa and Orobanchi minor. Next, please. Yeah. This is a carrot field infested by Orobanchi crenata. Orobanchi crenata is the largest Orobanchi, and plant height would be around 1.4 meters. So we cannot see, but the this field was also infected by Feripanchi ramosa and Feripanchi aegyptiaca. Okay, when you have this, you cannot get a good carrot. Even you have some carrot, small carrot, the sugar content are quite low, so that you cannot commercialize it. Okay, next, please. This is a tomato field, although you can see some watermelon here, but the and on the right bottom is a flower of Feripanchi ramosa, branched broom rape. And this was in Italy. And according to the farmer who owned this land, he said that when he planted tomato for the first time, 20 years after he obtained this area, tomato production was heavily affected by Feripanchi ramosa. It means that the Feripanchi ramosa seeds are waiting for their preferred host for more than 20 years. Next slide, please. This is a picture of Orobanchi minor, or small broom rape, or clover broom rape in Japan. Orobanchi minor was first reported in 1937 near Narita Airport, International Airport. Of course, at that time, there was no airport, but after that, it spread almost all over the Japan, except for Kyushu Island and also Hokkaido Island. On the left, that was taken in near the Narita Airport. The middle is Tokyo, and the right is Utsunomiya, our city. Next slide, please. It's a little bit heavy. Okay. I would like to talk about the distribution of these root parasitic weeds. This slide shows the distribution of Striga asiatica. It's one of the striga, and originally they are found only in Africa, but it spread to other area of the world. In 1950s, early in the 1950s, it was found in North and South Carolina, and the United States started the eradication program, and after 60 years of eradication program, they could reduce the infected area by 99%, but they still have 1% of infested area. So of course, it was very expensive, and it took quite long. And it spread to other countries. And recently, it was found in Queensland, Australia. Okay. Next, please. Next slide, please. This is a distribution of Striga helmontica. So far, this root parasitic weeds are found only in African countries, sub-Saharan African countries. Okay. Larger symbols indicate, as you can see in the bottom, the larger symbol indicates countries in which it causes significant crop losses. Smaller symbols, mainly they parasitize only wild host and cause lesser crop damages. Next, please. This is distribution of Striga gesnerioides. This is another type of Striga. It is a little bit different from other Striga, Striga asiatica and Striga helmontica, because this Striga parasitize on dicot, mainly legumes. So yes, it spread to Asia and also into Australia. Okay, next, please. This is a distribution of Electra. Electra is very similar to the Striga gesnerioides, but so far it can be found only in African countries. Next, please. This is a distribution of Robanchi crenata. If you can remember that the largest Robanchi, and it's very large, and open circle means that the circle indicates other countries with potentially suitable climate for its growth. And indeed, this weed, Robanchi crenata, was found in UK, United Kingdom, very recently. Next, please. This is Orobanchi cumana, sunflower-specific broom rape. It's parasitized only sunflower. And as you can see, all of the Orobanchi species originally found or distributed only in the country, Mediterranean areas, and it spread to other parts of the world. And recently, it invaded China, and where it causes severe damage to the sunflower production. Okay, next slide, please. Excuse me. This is a distribution of Felipanchi ramosa and the Felipanchi aegyptiaca. They are very similar. And yes, as you can see, that it spread to the world. And you can see them distribution in North and Middle and South America. And also recently in Australia, where it has been some eradication program is going. Next, please. Okay. This is a newsletter of International Parasitic Plant Society. hostrium. Hostrium is the name of the special organ of parasitic plants. It's connect parasitic plant to host plant. So parasitic plant takes nutrient and water through this hostrium. Okay, so it's the name of the newsletter. And you can see that in the press releases, click please, one click please. One click, please. Or,
Okay.
yes. One more, please. Okay. Decade-old weed seeds trigger new plants parasite outbreak. It said that in the early 1980s, devastating parasitic weeds was found in tomato field in California. It said that it's a branched broom rape, Orobanchi ramosa. Now we call it Felipanchi ramosa. Was treated aggressively, and the field was quarantined to prevent further spread. But when tomatoes were planted in the same spot more than 2 decades later, the branch boom rapes quickly returned. This means that the 20 years is not enough to reduce the seed bank of this root parasitic weeds. That's a big problem. So once you have these root parasitic weeds, it's rather difficult to grow potential host crops at the same location. Next, please. This slide shows the life cycle of root parasitic weeds. There are some differences among root parasites, but they largely the same. They produce the huge number of very tiny seeds, and the seeds will not germinate until they receive some signals produced by and released from the host root. We call, one click please, we call this click please, yes, germination stimulant. One more click please. Yeah, after germination, there will be involvement of the secondary signal from the host root. It's the radicle elongate attached to the host root and then penetrate into the cylinder, central cylinder, and connect to the central cylinder and establish parasitism. The root parasite grow underground for, it's depend on the species, but for several weeks to several months, and then produce flowering shoot or scape, and then set flowers, and so on. There's a host-dependent life cycle of root parasitic weeds. Next, please. This is a typical, this slide shows a typical comparison of the size of the seeds of root parasitic weeds and some crops. It's a rice. This is the seeds of Orobanchi minor. Okay, you can see the difference of their size. A single root parasitic weeds produces 10,000 to 500,000 of dust-like seeds, which remain viable for many years, more than 20 years. A few root parasitic weed plants per square meter maintain their seed bank. So once you have this, it's quite difficult to eradicate them. Okay, next please. Because I'm a natural product chemist, I have been working on the germination stimulant. It has been known for a long time that there are some chemical communication between root parasitic plant or root parasitic weeds and their host. Because root parasitic plants live in the vicinity of their host, a host root exudate induce parasite seed germination, And indeed, Brown and co-workers in mid-1950s collected active root exudate and partially purified germination stimulant. They concluded that most plant species produce stimulants. Stimulants can be absorbed by activated charcoal, activated carbon, and stimulant contain lactone group because they are stable in weak acid but unstable in basic condition. One click, please. Lactone group, as you can see in this slide, it's a intramolecular ester. Okay. Next, please. As you can see in this slide, actually, the germination stimulant contains the lactone groups. Okay, on the left top, this is the Strigo, the first isolated germination stimulant for Striga. It was isolated from the cotton root exudate in 1966. And along with its acetate, acetate, excuse me. And it contains A, B, C, D ring, 4-ring system. And but at that time, cotton is not a host of Striga. So some people complain that the Strigo would not be a genuine germination stimulant for Striga. But in 1992, sorgolactone was isolated from the Striga genuine first, sorghum. So, and also in 1992, Alexrol, this structure is not correct, but anyway, it was isolated from cowpea root exudate. And in 1998, we isolated this compound named orobanco because it's induced germination of orobanche, and also it contains hydroxyl group, so it's an alcohol. So we named orobanco. That's why this was isolated from red clover root exudate. It has been known that the strigol also not only induce germination of Striga species, but also orobanche and Alectura. Okay, one click, please. Yes, as you can see, all of the Striga lactones contain the D-ring moiety, the same D-ring moiety. At that time, when we started working on these Striga lactones, there are no analytical method to detect this compound because the plant produce only minor amount, very small amount of strigolactones, and also strigolactones are chemically unstable. Okay, next slide, please. But fortunately, in 1990s, a new technology named liquid chromatography tandem mass spectrometry became available, LCMS-MS. So by using this machine, we can analyze selectively and with very high sensitivity these strigolactones. Now, one click, please. One click, please. Yeah. Yes. This LCMS-MS is now a standard method for pesticide residue analysis and doping test. Very sensitive and specific. And this analytical method can be conducted without any, for example, the derivatization. This means that for other analytical method, you may need to change the structure of the compound to be able to detect by that method. But the LCMS-MS, you don't need to do that. So it is important method. Now, next slide, please. These LCMSMS has been used in analysis of stride-reluctant, also in some other plant hormones or plant-derived chemicals. For example, this is a 3-channel MRM chromatogram of Pisum sativum. It's a garden pea. Okay, by using different channels, we can detect orobancol, electrol, and 4-deoxyorobancol. And in this method, we can use what we call the column chromatography, by which we can separate these compounds. These compounds come out from the column at different times. So you can distinguish these compounds, and also by the MRM, you can confirm the presence of each stride-reluctans. Next, please. And in 20 years, the machines have developed very significantly. And these 3 stride-reluctans are very similar in the structures. Only difference is the position of hydroxyl group. Okay, strigol, sorgomol, orobancol. And as you can see, top and the middle, the retention time. So we call the retention time, or when this compound come out from the column, the same retention time. And the bottom one is slightly fast, but the very almost the same. But by using the different channels, we can distinguish these 3 stride-reluctans very easily. And now, so far the stride-reluctan would be around 40. So we can analyze and quantify these stride-reluctans, nearly 40 stride-reluctans within 20 minutes by using LCMSMS. Okay, next, please. Okay, so I shortly explained about the structure. We don't need to care about what the structure is, but the stride-reluctan can be divided in 2 groups depending on the structure. Okay, one is the canonical stride-reluctans, which contains A, B, C, D ring moiety. Okay, these compounds we call canonical stride-reluctans, and also that the canonical stride-reluctan can be divided into 2 types according to the orientation of the C-ring. In this strigol type, the C-ring is coming up from this plane. Okay, it's coming up. And you can see that in the strigol type, they have different hydroxyl group at a different position. And also, next, please. Next, yeah, this is orobancol type. You may remember that the C-ring is now it was drawn by a dot. It means that what we call this alpha oriented, well, it goes downward from this plane. Orobancol type have many derivatives, but all of them have the hydroxyl group at the same position. So it's slightly different from the strigol type. Okay, next, please. So these are non-canonical stride-reluctans. Okay, for example, heliolacton contains only A and D ring, and also the next one also contains A, D rings. And the Z-lactone is from Z maize, corn, maize, only D rings. Okay, but the bottom abenol contains A, C, D rings, and also canalactone is from cannabis, S, A, C, D ring, and biosymbiol recently identified from various plant species is A, C, D rings, and lotus lactone, A, B, D rings. So these non-canonical lactone lack one of the rings. Okay, next slide, please. So in addition to the strangulactones, there are many chemicals released from the plant roots, and these chemicals are involved in the communication between plant and other organisms, including parasitic plants, weeds, bacteria, nematodes, fungi, and so on. Next, please. So it has been a long question, sought question: why plant produce and release strangulactone, which induce their enemies, germination of their enemies, root parasitic plant. And in 2005, it was found that the plant produce and release strangulactones to promote the symbiosis with avascular mycorrhizal fungi, AM fungi. More than 80% of land plants form symbiotic relationship with AM fungi. By this symbiosis, plant can obtain nutrient like phosphate and nitrogen, and also water. In turn, AM fungi receive photosynthase and fatty acid, and so on. Next, please. Next, please. This slide shows the effect of avascular mycorrhizal symbiosis. On the left, you can see the growth of plant without symbiosis. Okay, so their growth depends on the concentration of the phosphate. So at low phosphate concentration, the plant growth was very poor. But once you have symbiosis on the right, if you can compare, you can compare at the concentration of 0.1 millimolar, the plant grows very well. So the plant grow well under phosphate deficiency when they have symbiosis, because you can see that in this photo, these are the plant root, as you can compare the hyphae, the very fine hyphae. So very fine hyphae can reach phosphorus-rich spot in soil because phosphorus or phosphate hardly moves in the soil. So, and also they are pleasant as a spot because it's a residue of dead plant or dead animals. Okay, so symbiosis is very important for plants, especially when they come up from the sea to the land. Okay, next slide, please. But some plant species, so 20% of the land plant will not form symbiosis. So, but such a plant species still produce strangulactones. Why? Because they have their own functions important in the plant. So in 2008, it was found that strangulactons are novel class of plant hormone inhibiting shoot branching. So in addition to their function as a fire branching in fungi and germination stimulation of root parasitic plant, they strangulactons important plant hormone. Okay, next please. So on the left, you can see the rice. WT means a wild type rice. It's a normal rice. And the D10 is a mutant, which cannot synthesize strangulacton. So they are dwarf, short and with many tillers. So when we add the synthetic strangulacton GR24, they grow normally. Okay, on the right, it's Arabidopsis, and the WT is a wild type, and also the MAX4 is a biosynthetic mutant. You can see the many branches, but when we add to GR24, phenotype is rescued. Okay, next please. So because strangulactons are important as a signal for symbiosis and root parasitic weeds, and also in plant as a plant hormone, the production and the excitation of strangulactons are important, seems to be regulated by various factors. The first, the nutrient availability, especially the phosphate and nitrogen, because mycorrhizal symbiosis can be found in more than 80% of land plants, and rhizobia symbiosis, yes, legumes, and other bacteria, it's endophyte. So these affect the production and the excitation of strangulacton. And also environmental conditions, light temperature, soil water content affect production and excitation of strangulactons, and also growth stages, especially the root vitality, affect strangulacton production. And recently we noticed that the plant roots sense strangulacton in the soil, and so it's some kind of a feedback inhibition was observed. Next, please. In this slide shows the effect of nutrient on the production of strangulactons. In red clover, because it's a legume, it can obtain nitrogen from rhizobia symbiosis. Okay, and so we examine the effect of nutrient deficiency on the production of the orobancol, one of the major strangulacton in red clover. One click, please. So only the low phosphate condition enhance promote the production of orobancol. Okay, next, please. By contrast, in sorghum, which will not form the rhizobia, both nitrogen and the phosphate deficiencies enhance production of 5-deoxystrangol. It's a major strangulacton in sorghum. So this, you can see the differences, different reaction of the strangulacton production to the low nutrient conditions. Next, please. So we examine the So different plant species and their response to the nutrient availability on the strangulactone exudation. One click, please. One click, please. Example, in red clover alfalfa, as I explained, low phosphate enhanced strangulactone exudation because they can obtain the nitrogen from the root nodule bacteria symbiosis. And in the case of tomato, tomato obtain nitrogen probably from the endophyte, okay? And so others, for like sorghum, both low nitrogen and low phosphate enhance strangulactone exudation. But there are some plant species, 20% of land plants, they will not depend on the M symbiosis. One click, please. One click, please. Yes. Neither low phosphate nor low nitrogen and has strangulactone exudation. Okay. Next, please. This is the time course of strangulactone production by garden pea. I forgot to mention about that, but all of the plant species examined so far produce at least two different strangulactone at the same time. Okay. In this case, pea produce 1, 2, 3, 4, 5 different strangulactone. So they release mixture of strangulactones. Okay, so and in the case of root parasitic plant, the seeds seem to be germinated after maybe 2 to 3 weeks after planting the pea. So in that case, herbacil acetate would be important strangulactone in inducing germination of root parasitic weeds. Next, please. Next, please. Next, please. Okay, and also that strangulactone profile seems to affect the host recognition by root parasitic weeds. This is the Orobanche cumana sunflower specific bloom rates, and from sunflower, 2 different strangulactone or the germination stimulant were identified. One is dehydrocostoslactone. It's not a strangulactone. And heliolactone, it's one of the non-canonical strangulactone from sunflower. Okay, before that time, we examined the effect of the different strangulactone on the germination of Orobanche cumana and also the mixture. Okay, we compare the germination response of Orobanche mana. It's Orobanche mana has a very wide range of hosts, so it parasitizes various plant species. And the Orobanche cumana is specific to sunflower. Okay, Orobanche minor responded very well to all of these strangulactones, stragol, orobancol, 7-oxyl, and also the mixture of them. By contrast, Orobanche cumana, for Orobanche cumana germination, stragol is not so good, but the orobancol is good, and especially the 7-oxyl orobanche acetate is a very best germination stimulant. And when stragol was combined with orobancol, the effect of orobancol seems to be reduced by stragol. So stragol shows some antagonistic effect, okay? And it was our surprise that the orobancol, it's a good germination stimulant, and 7-oxyl orobanche acetate, the best one when we tested, we could not see any germination. So there is very clear antagonistic interactions. This can be found in other experiment, another experiment. Next slide, please. This slide shows the germination stimulation by plant root exudate from different plant species. Of course, on Orobanche cumana, of course, the sunflower root exudate induced very high germination of Orobanche cumana, and also the cotton, which produces strigol and strigal acetate, not good germination stimulant. And GR24 is a synthetic strangulactant. Okay, although we know that the legumes produce good producer of orobancho and also the 7-oxyorobanchol acetate, you can see that they are very weak or almost no activity was found in root exudate of legumes. This means that although they produced strangulactons, there are significant antagonistic effect among the strangulacton produced by legumes. Next, please. Now, I would like to change to the control measures. Yeah, there are many possible control measures. The one is breeding of resistant cultivars, and I will explain this later, and crop rotation. But the crop rotation is rather difficult because small weed seeds with long viability. So once you have that, the seed of root parasitic weed is quite difficult to grow potential host crop, okay? And hand and mechanical weedings, because root parasitic weeds attached to the underground, so it's impossible to detect at early stage of parasitism. And also they attach to the host root firmly, so it is very difficult to remove them without damaging the host root. Okay, the next, soil fumigation. Methyl bromide is very effective, but already banned. And suicidal germination, I will explain this later. And the herbicides, herbicide-resistant cultivars, a non-GMO is a good strategy, and biological controls are also good strategy. Pathogens, herbivores, toothpicks, kill, and cultivation method, push-pull. Okay, I will explain this. Next, please. This is a very famous Professor Gebisa Ejeta. He is originally from Ethiopia and has been working in Purdue University in United States. He has been working on the breeding of striga-resistant cultivar of sorghum. And he had a lot of work and selected striga-resistant cultivars, and he obtained the 2009 World Food Prize. It's a Nobel Prize in agriculture. Okay, and recently, one click, please. Why the resistant cultivar he selected is resistant to striga has been elucidated because one of the genes they termed a low germination stimulant 1 mutation of this gene change the kind of strangulactons in susceptible cultivar to different strangulacton in resistant cultivar. And the susceptible cultivar, the major strangulacton is 5-deoxystrigol. And it was changed to orobancho. Orobancho is rather poor germination stimulant for striga. So by changing the type of strangulactone produced, it becomes resistant to striga. Next, please. Soil fumigation. So it is, it is possible to kill the parasite seed in the soil by fumigation with methyl bromide, but it's not allowed now. And Dazomate releasing methyl isothiocyanate can be used as a soil fumigation, and solarization covering by vinyl or plastic films, and kill the seed by the heat of sunshine. Okay, and suicidal germination. Suicide germination is inducing seed germination in the absence of host. Actually, in the United States, striga eradication program was done by using the ethylene fumigation into the soil. Some of the striga, not all, but the striga asiatica and the striga helmontica seeds can be induced to germination by ethylene. It's a gas. Okay, so by using the ethylene repeatedly, ethylene fumigation into the soil repeatedly, they can eradicate the striga in the United States. And Dazomate can be used to induce to kill the seed, and also it may induce seed of Peripanchyramosa, one pathotype of Peripanchyramosa, which parasitize oil seed lapes. And this was used in Australia to eradicate the Peripanchy over there. And also there have been a lot of work have been done by develop strangulactone mimics, which induce germination of striga seeds. But some people was worried about that it may promote parasitism. Next, please. And yes, herbicide-resistant cultivars. Okay, root parasite depend on the host plant for supply of water and nutrient. It's a very strong thing. So a herbicide safe to crop plants are rapidly metabolized in the host and not effective to parasite. And herbicide-resistant cultivars can be selected for some herbicide, AHAS and HCS inhibitors. AHAS is the key enzyme in the branched chain amino acid, and the HCS is a key enzyme in the fatty acid biosynthesis. It is interesting that although root parasitic weeds depend on the host for the supply of water and nutrient and some other compound, they need to synthesize synthesize branched amino acid and fatty acid by themselves to some extent. And resistant. So these resistant cultivars are not transgenic. Okay, and seed treatment or seed coating with herbicide also control other weeds. Next, please. Next, please. This was done in collaboration, CIMIT in collaboration with Weizmann Institute. And in this, you can see the picture, it's the imazapyl. It's ACCAs, AHAS inhibitors. So by using this and its resistant cultivars, they can control the striga, okay, effectively. Next, please. And biological controls. The pathogen of parasitic would be specific, and an extensive study has been conducted to develop parasitic with specific biocontrol agent, fungi, bacteria, and so on. Most fungi pathogenics to parasitic weeds have been found to produce trichosethines. It's a highly toxic compound to animals and human beings. So you cannot use this fungi, okay? And in general, pathogenic microbes are difficult to culture, and 99% of soil microbes are unculturable. Difficulties in distribution and storage and inoculations. Okay. Next slide. And recently, yeah, probably you all know that about this, the Kenya Kichawi kill have been developed, and yeah, it's a first bioherbicide to combat striga. A fungal pathogen was isolated from wilted striga. The pathogen induced overproduction of specific amino acid only in striga, and also that it can grow on the toothpicks and on the cooked rice, and then by inoculation is also easy. Okay, on the left picture, you can see the difference and the effect of this Kichawe kill. Okay, next, please. One more click, please. So next is a cultural method called push-pull. This is originally designed or developed to reduce the damage caused by the insect. It's a stem borer. Okay. So the pull means that in this cultural method, maize plants were surrounded by Napier grass, and also between the row of maize, a legume, Desmodium, are planted. Okay, the Napier grass produce volatile chemicals which attract moths to lay eggs, and push mean that the Desmodium produce volatile chemicals which repel mosses. Okay, so this cultural method, but this method is also effective in reducing damage caused by striga. Next, please. Next, please. Okay. And because of the Desmodium, it's a legume, produce strigolactone. It's induced germination of striga. But as I explained before, striga cannot parasitize dicot. So it's some kind of suicidal germination. And also, Desmodium at the same time produce some chemicals which inhibit the growth of striga radicle, so that it inhibits the germinated seeds to attach to the host, in this case maize. Okay, by this push-pull effectively reduce the damage caused by insect and also striga. Okay, next, please. Next, please. So we need to develop new control measures continuously because, for example, breeding of resistant cultivars, we have seen, for example, in the case of sunflower, when you have the resistant cultivar, the parasite will evolve the new biotype which attack the resistant cultivar. And yes, crop rotation, we need to find some good crops, and hand and mechanical weeding would be important to reduce removing the resistant biotype. For example, resistant biotype will be evolved in the next, for example, the herbicide-resistant cultivars. Okay, so it is important. And soil fumigation, suicidal germination needs to combine with following. Okay, and herbicide-resistant cultivars plus herbicide, it may have the risk of evolution of resistant biotypes. So you need to change the maybe the crop and the herbicide, okay? And by contrast, biological controls may have the lower risks of such resistant biotype evolution, and push-pull also lower risks of such resistant biotype evolution. Okay. Next, please. So that's all. Thank you for your kind attention. I'll be very happy to have any comment and questions. Thank you very much.
Okay, thank you very much, Professor Yoneyama. So please give it a sign of the applause, everyone. Thank you very much.
Thank you.
Okay, so we'd like to have this comment from the Professor Wesenga, please.
Yeah, thank you very much.
My pleasure.
Thank you very much, Professor Koichi, for this nice presentation.
Thank you.
And I'm happy to give some commentary on the presentation. In my commentary, I want to break it into two sections. One, I will look at the content of the presentation. I will also talk about the context and the relevance of the presentation, and perhaps I will give some recommendations based on what I have listened. And in terms of content, Professor Koichi has taken us through a number of aspects regarding parasitic weeds and focusing mainly on striga and broomrape.
Yeah.
He gave us a definition of parasitic weeds, their classification. We're giving examples such as the Doda, Cascuta. And just at this point, I would like to mention that in Kenya, we have quite a wide spread of Cascuta within the countryside. So if you drive around, you're going to find Doda Cascuta infesting a number of plants. And perhaps this would be something quite interesting for us. He gave us the different types of the parasites, the holoparasites, the hemiparasites, and the facultative ones. He went to give us mention issues in biology has to do with reproduction, especially the issue of seed production and their persistence in the soil, which enables the weeds to be a problem over a very long time. Mentioned the importance of strife and the economic importance in the sub-Saharan Africa in terms of economic yield losses. He went ahead to discuss the diversity and distribution of striga and orobanche, the different species, and sometimes there. He went ahead to give us the distribution of these species globally, and discussed the importance of seed bank in the infestation. He took us through the life cycles and especially the role of chemical stimulants in the life cycle of this, including their role in the establishment of parasitic relationship with plants. And he took us through chemicals that are involved, the strigol lactoses, and giving us the methodology for their detection, the most preferred method. He mentioned the link between the strigolactose and the AMF fungi, which is really the basis for why these chemicals are being produced. He took us through the mineral nutrition and how they interact with the production of the strigolactones. Then he took us through the chemical, control of these parasitic weeds, giving us some very practical examples, including the Kichawi kill, which is currently being practiced in western part of the country, as well as push-pull technology, which I think some of us are quite familiar with. So it's quite a rich presentation in terms of content. I think it provides a good basis to understand these parasitic weeds, their importance, and how they could be managed. So the science behind some of the management practices that we have. So it's a very rich one. And Professor Koichi, I think, presented in a way that anybody could actually understand. I could follow the presentation quite well, which is quite good. Now, what about the context and relevance? This presentation comes at a time where I've already mentioned in Kenya, many of these parasitic weeds, I think we can easily relate with them. Weeds like striga are a major problem in our country, causing serious yield losses. Within sub-Saharan Africa, if you look at the CIMIT website, you will find indications of infestation of up to 20 million hectares of land infested by striga, with yield losses of value up to $1 billion and affecting 100 million people. In East Africa, it's estimated that 1.4 million hectares are infested by striga. In Kenya alone is 340,000 hectares. So you can see if we are talking about food security, this is quite important. We need to address these weeds so that we can at least maybe become food secure. Managing these weeds will play a major role in addressing or meeting at least the SDGs, especially SDG number 2, that seeks to attain zero hunger. And this is quite relevant for us. I've already mentioned a weed such as Doda is already a big problem in Kenya. So I think this presentation comes in quite handy. It comes in at the right time. And I think it helps us to think of some of the ways that we could try to manage some of these weeds. Now, for me, some of the take-home messages that I can quickly mention or make some of the recommendation has to do, I think it is in line with the entire program that we are talking about, building human capacity. I don't have exact figures, but generally we have very few weed scientists in Kenya, and perhaps that would also be true for the rest of Africa, maybe even globally, I don't know. We don't have as many people. If you compare, for example, entomologists, pathologists, you have quite a number of them. But when you talk about weed scientists, I think they are quite a handful. So what I think is we need perhaps, I'm hoping that through this kind of interaction, we can motivate young scientists to pick up some of these area of weed science so that we build human resource capacity. Perhaps with a professor like Professor Koichi now is emeritus. As he retires, perhaps he can participate in building a human resource capacity that can help us continue with this important research. Because as you can see, these weeds are quite problematic. And if we do not address them, perhaps we may not be able to meet some of the developmental agenda that we have. And maybe one of the areas that I think would be interesting to look at, perhaps, I don't know, how does that fit into the changing climate? We now have climate change as a major problem. I think at some point, Professor Koichi mentioned that there are some factors that are involved in this production of these chemical stimulants. And one of the factors that affects them would be the environmental conditions. And if these conditions are changing because of climate change, perhaps we need to be looking at how do the expected climate change affect the these chemical stimulants, their production and elucidation, and how can we better prepare ourselves to deal with maybe the new weed problems that are going to emerge as a result of climate change. I think the other whole question to me would be, how do we design our management practices? Because I think some of the problems we end up with weeds has to do with the way our cultural practices, which disrupt maybe the ecosystem within which we are growing our plants, and ultimately that results into weed problems and other pest problems. So how can we utilize the information, the knowledge that is known, the science, and then we are able to design management practices, not just for weeds, but for growing our crops so that we do not have these weed problems. Thank you very much for the opportunity to comment on Professor Koichi's presentation. It was really a pleasure to listen to you, Professor Koichi. Thank you.
Thank you very much, Professor Wesonga. It is a very nice comment. Professor Yoneyama, do you have any especially about the effect of the climate change to the damage of the witchweed to the agricultural crops.
Mm-hmm. Okay. Yes. As I didn't talk about much about the Cuscuta. We do have Cuscuta in sea area, very close to the sea in Japan, and it is now spreading in Japan. And so climate change will have big impact on the agriculture worldwide, and also they will affect the distribution of, for example, root parasitic weeds, especially the striga. And so far, we don't have any striga species in Japan, but we may have striga because it may be much drier conditions and hot. So that is good for striga species to survive. And recently, of course, the weed can be weed when you have the agriculture. So in agricultural area or agricultural field, some plant species adapted to that condition where agriculture was conducted, they become weedy. So, and also they are changing. So for example, recently, the striga species has been found to attack wheat. Okay, so another important crops. So we may have the effect of infection of wheat by striga species in the near future. And I would like to strongly recommend, especially the young scientists in Kenya, because next year we will have, or actually I will not attending, but the International Parasitic Plant Society meetings will be held in Kenya. So it was postponed by, according to the COVID-19, but next year it will be held in Kenya. So I would like to encourage many young scientists to attend the meetings. Okay, that's all.
Okay, thank you, Professor Yoneyama. I'll also give this a nice announcement. It's the International Congress will be held in Kenya. Okay, okay. So now we'd like to accept some questions from the audience. Anyone have a question? Please raise the hand using the Zoom function. Now let's see in the any comments? Oh, maybe in the chat or also. Oh no, that's obvious. I see. I'm going to stop with that. Okay. Okay. There anyone have okay, John? Professor Wesonga, please.
Yes, if there are no questions, I can still ask. Maybe just an interest, like I mentioned, I think our resource capacity in Kenya, particularly in respect to weed research, I think is not that high. What I was wondering is whether there are any opportunities for collaborative research, especially working with Professor Koichi. I don't know which opportunities, if there are any, that we can probably consider.
Okay. Okay. Thank you very much, Professor Wesonga. I'm not sure that Professor Yoneyama can have my help with that. I don't know. How do you think, Professor Yoneyama?
So actually, that many, for example, scientists, Japanese scientists, including some scientists at Nagoya University, some groups are working in collaboration with Kenya that they are doing. They're trying to do the suicidal germination by using their own chemicals. It's very effective. That compound was reported in Science, and unfortunately, I met him, one of the scientists at Nagoya University. They tried to infect crops by striga, but there was no infection by striga in their plot, so they failed. But this year they will conduct another field trial to see the effect of their chemicals as a suicidal germination inducer. Okay.
Thank you, sir, Professor Yonema. Are there any the questions or comment?
I have question.
Okay, go ahead.
Thank you for the presentation, and it was nice, and I'm interested very well, especially on the distribution map on the parasites which are found in the globe.
Mm-hmm.
It's very interested. I'm in Ethiopia.
Uh-huh.
So currently I know where is these plant parasites distributions are found, and here in Ethiopia, maize and sorghum, wheat are very common staples for in Ethiopia. So I found the distribution of that parasites which affect our staple common food. That's a very interesting one. And I found the question is in the mitigation or in the measure that mitigate to reduce the parasitic attack. There is one thing, methane where is it? Soil fumigation. In the soil fumigation, methyl bromide is not.
Mm-hmm.
It's not forbidden. You said it's not prohibited. Why it is? Is that due to the environmental problem or what's the problem? Is that? That's my question.
Yes, of course. It's destruct the methyl bromide destruct the ozone layer. So it was forbidden except for some quarantine area. Special, they need permission to use that.
Okay. Relatively to its cost, is that?
So of course it's expensive also, and but because it's destroy the ozone layer, it has been forbidden to use, except for some restricted area. Okay.
Okay, thank you. That was my question.
Okay, thank you to your questions. Maybe one more question. Does anyone have questions?
Okay, thank you. I have one question.
Mm-hmm. Go ahead.
I'm Victor from Meru University, Kenya, a master's student in horticulture. And thank you, Prof, for the presentation. It's quite interesting because sometimes back in our area, I was seeing this striga weed affecting the maize plantation, but it was quite funny that where there was an intercrop between the legumes and the maize. The maize was the one which was affected, but the legumes was not affected much by the weed. And my question might be is that what about if we can intercrop the legumes and the monocots? Can it help to reduce the spread of this trigger weed? And also another question that I might ask is that is there any mutual relationship or the antagonistic relationship between the parasitic weeds and the soil microorganisms like the mycorrhiza? Yes, thank you.
Okay, so one point that because striga, except for striga gesnerioides, striga helmontica and striga asiatica will not parasitize on dicot. So they will not parasitize on Desmodium or legumes. Okay, so of course the legumes produce strangulactone, so their root exudate induce germination of striga, but they will not germinated striga cannot attach to the to legumes. So that's the point. And also there are many reports that when you have the microalgal colonization, production and release of strangulactone reduced because they have enough phosphate supply. So as I explained, that the strangulactone production and exudation is enhanced under nutrient deficiency, especially phosphate and nitrogen. But when we had the avascular microalgal colonization, you have enough phosphate. So the plant reduce release production and release of strangulactone, and then reduce the germination of striga seed. So it may help. And also by fungi colonization will protect or will enhance the vitality of the crop. So they are a little bit more tolerant to striga. If they attach, they become a little bit tolerant if they have the M fungi. Okay.
Thank you.
My pleasure.
Okay, thank you for your question. There is one question in chat. I read it out of this question. Thank you for your presentation. Some mixture of strigolactone enhances, but the other inhibits germination of striga. What happened chemically in soil?
Other inhibit germination of striga. Okay. We haven't actually tested the mixture of strigolactones on striga seed germination, but probably striga helmontica and the striga asiatica, the seed of these striga species can be germinated in any mixture of strigolactone. So that Robanki kumara is very specific. There are some strict or antagonistic effect observed only for that. But I don't think that striga striga species we can see any antagonistic effect of strigolactones. And in the case of sorghum, resistant sorghum produce orobancol. It can induce the germination of striga, but it is rather weak germination stimulant. So as compared to the 5-deoxystrigol, the major strigolactone produced by susceptible strigolactone. Okay. Also, they produce other strigolactones, but I don't think that there are any antagonistic effect among the strigolactone in the case of striga germination stimulation. Okay. But we haven't examined all possible, for example, mixture. So because it's only limited, so we cannot examine all different types of mixtures of strigolactones on germination of striga species. Okay.
Okay, thank you for your answer. I think it is Koyama-san is that is answer is will be okay. Says thank you. Okay, thank you. Okay, let's it's almost past the 7. So please give this applause to the Professor Yoneyama. Thank you very much, Professor Yonema.
Thank you. My pleasure.
Okay, so I'd like to give this announcement. So give me an push. Okay. Hmm? あれかんないな。Okay, and okay, this one. Okay, this oh, give me the sorry. Okay. This is only become a little bit bigger. Okay. Okay, this is the next symposium will be held is December 23rd of this year. It started from 5:30 in Japan time, 11:30 from East African time, and 8:30 in Ghana. So there is at the bottom there is the application site. I hope there's many people will again attend this symposium. And also we have this fourth symposium, which will be held in January 28th next year. Also, there is silent here. I hope also many people also go attend this fourth symposium. Okay, and also we would like to have the post question of the second year international symposiums here. That is shown in this slide. Please answer some of the question. We will be very grateful. Okay, this thank you everyone for joining the second UA series symposium. That was a very nice symposium. Thank you very much all for your attendance and the cooperation. Thank you. Okay, let's bye now. Okay, thank you. Bye-bye.